Hi, everyone. Can you hear me all right? Okay, great. All right, so as Lisa mentioned, I'm an epidemiologist, and epidemiology is the study of disease patterns in the population. The work I do and that I'm going to talk about mostly focuses on how genetic data informs those patterns, specifically patterns of autism. And I've been working on this for about 6 or 7 years now, initially starting with some twin studies, but building up to the types of measured genetic variables that I'll be discussing today, and pretty consistently focused on ASDs. So who here has some familiarity with autism or autism research? Okay. And how many of you are here specifically because of an interest in ASDs? Right, great. So for those that did not raise their hands, we have the introduction here. ASDs, autism spectrum disorders, include a very heterogeneous and large group of neurodevelopmental conditions that are characterized by a small set of core features. Social and communication deficits, and those can take many forms. It can involve things like not making eye contact or not speaking at all. There's a wide range. And restricted and repetitive behaviors and interests or activities. That can range from everything from hand flapping, which would be a behavior, to an interest like a very consistent interest in something like trains. Now, most people, regardless of whether or not they have an interest in autism, have probably heard that autism is diagnosed much more frequently now than it was 20 to 30 years ago. It's in fact diagnosed probably about 20 to 30 times as frequently as it was 20 to 30 years ago. And there's no clear way to say exactly why that is the case. What we can definitely say is that the majority of that increase reflects diagnostic changes and increases in awareness. So one of the easiest ways to see that and think about it is that there's an obvious shift in certain things, certain properties of the medical comorbidities and diagnostic comorbidities of the diagnosis that have changed over time. So about 30 years ago, one would expect 70 to 80% of people diagnosed with autism to meet criteria for intellectual disability, which used to be referred to as mental retardation and generally involves having a measured IQ below 70. So 30 years ago, when autism was markedly more rare, 80+% of people with a diagnosis met criteria for intellectual disability. At this point, that number is under a third. So there's been a seismic shift in the average conceptualization of ASDs in the US and Western European populations mostly, which has resulted in a shift towards diagnosing into what is often referred to as the higher functioning range. So whereas 20 to 30 years ago, there was more consistency in what you could expect in terms of outcomes from those who were diagnosed and that they were more severe. Now, in the US and in Western Europe in particular, people who are diagnosed with ASDs can be dependent on constant care for their entire lives, but they can also grow up, be independent, have families of their own, and be extremely professionally successful. There's a tremendous range. And that range is in no small part a consequence of the diagnostic expansion, which is the primary driver of the prevalence increase in ASDs in the US population. In the context of all these changes, it's not actually possible to say whether there might be a real increase. It's just not something that one can readily determine. So what I'm going to be talking about is some changes in the epidemiology of autism focusing on these diagnostic trends and shifts and then relating them back to genetics. So I mentioned the time progression in terms of what you see in ASDs with regard to the comorbidity of intellectual disability. But even within the United States, there's still a lot of variation in the prevalence of autism. And you can see from various data sources that a lot, if not the majority of that variation between US states, even at this point in time, comes from the extent to which those states have embraced the diagnostic expansion. So for example, if you look at New Jersey, which has the highest autism rates in the United States, there's about twice as much diagnosed ASD in New Jersey as there is in, let's say, Arkansas. The red bars here, elegantly prepared by Emily Wigdor in my lab, show the rate of autism with intellectual disability in that state. The blue bars show autism without intellectual disability. So if you look at New Jersey and Arkansas, you can see that the rate of autism with intellectual disability is pretty much the same. And the reason you see that twofold difference in prevalence is because the rate of autism without intellectual disability is vastly different. So this change we've seen over time in terms of changing diagnosis is relevant within current diagnostic practices across US states. It's still the major driver of prevalence differences in ASD. So with that introduction, let's talk about some genetics. Neuropsychiatric disorders in general are very heritable. Heritable means that they are influenced by genetic variants. And there are lots of different ways for estimating how much genetic variables matter to one's risk of a disease. Classically, people used twin studies, and the classic twin approach is to compare how similar monozygotic or identical twins are to dizygotic or non-identical twins. Since monozygotic twins share all or virtually all of their genetic variants, we expect any trait that's influenced by genetic differences to be more commonly shared by identical twins than non-identical twins. And you can see that for lots of different traits. This is an example from schizophrenia from a very long time ago, where you see more similarity between the identical than the non-identical twins. Now we have lots of different ways of estimating heritability, and across them, you consistently see that disorders like autism and schizophrenia and bipolar disorder are highly genetically influenced. And they're as genetically influenced or more than a lot of diseases that we typically think of as having strong genetic predispositions, like type 2 diabetes or breast cancer. Now, in trying to figure out which types of genetic differences might influence autism risk, we also pay attention to a second pattern, which is that people with schizophrenia and autism, this is an autism talk, so we'll talk about this one, people with autism have fewer children on average than people without, and that's called a reduction in fecundity. So men with a diagnosis of autism have about 75% fewer children than men without, and women with a diagnosis of autism have about 50% fewer children than women without. And this reflects functional impairment and lots of other things that comes with the disorder. But it also tells us something about the types of genetic variables we can expect to actually influence autism risk. Specifically, when you're thinking about a disorder that is associated with a loss of fecundity, particularly a strong loss of fecundity like this one, genetic variables that have a large impact on risk, create a lot of risk, are rapidly pushed out of the population by evolution. So let's say, you can kind of imagine this intuitively. If there's a genetic variable that gives you a 50% chance of developing a disorder that reduces the number of children you have by 75%, that variable is going to rapidly be pushed down in frequency in the population because it's much less likely to be passed on to future generations. So from that, we have two types of genetic differences between people, which I'll be calling variants, that we think and have found to be particularly influential in creating genetic risk for ASDs. The first is common variants, those that are seen in virtually all of us in this room. And those, because of this evolutionary influence, have to have tiny little effect sizes. They create itty little bits of risk so that they can escape a rapid evolutionary filter. The second is large effect alleles that are new to a generation called de novo variants. And we'll talk about those a bit, both of them. So actually skip that one. So common variants in the population are just genetic variants that are seen in more than 1 or 5% of people, depending on your favored threshold. Like I mentioned, the genetic variants that are common that we've associated with disorders like autism and schizophrenia have very small effect sizes. For example, they increase risk by almost always, less than 10%, which in the context of a 1% condition means that having one of these things would take your risk from 1% to 1.1%, or much less than that. So each individual common genetic variant has a tiny amount of influence, but thousands or tens of thousands of them act together in aggregate over the entire genome and can give you a total amount of risk that is quite substantial if you add them all up. And we refer to this as polygenicity, where common variants act together, each contributing a tiny little bit, but they work across the genome to create risk. And that's not dissimilar to the genetic architecture, for example, height. There's not one gene for height. There's not 10 genetic variants for height. Genetic influences on height are spread across the genome and are very, very small. So a similar story with rare variation exists in that any large effect, rare variant, will be pushed out of the population over time. But the exception to this are things that we call de novo mutations. De novo mutations are genetic variants that are new to a person. They're not seen in either of that individual's parents. They're mutations that occurred in the formation of a sperm or an egg cell. Because those mutations are new to that individual, selection has not had an opportunity to act on them. So you can have a de novo variant with a very big effect on a very reproductively deleterious outcome, which gives us two main types of genetic differences to think about. The first is polygenic risk, like I mentioned. It's continuously distributed. It's the additive effect, the total across the genome effect of these tiny little influences. And polygenic risk for autism, schizophrenia, and any other trait you can think of is present in some degree in all of us. It's just a continuously distributed phenomenon. And then there's de novo variation. Virtually all of us also have de novo variants. On average, they're about 40 to 60 a person, plus or minus some wobble in those estimates. Very few of those de novo variants that people in this room carry create a sizable risk for ASD. So while most of us have, all of us, have de novo variation somewhere in our genome, it rarely, in people who do not have a neurodevelopmental disorder, less than probably 5% of the time, creates large amounts of risk for ASDs or another neurodevelopmental condition. So we have these two types of genetic risk factors that are associated with ASDs, and we're interested in linking that back to the types of differences in autism itself that I talked about at the beginning. So we know there's a huge amount of heterogeneity in autism. We know some of it relates to IQ, some of it relates to your ability to speak, some of it relates to whether you have seizures, some of it relates to functional outcomes. How do these different genetic variables map on to those differences in what we call phenotype? What's the relationship between genetic differences and phenotypic differences in people who have autism? So before we go there, I wanted to show you this because it kind of importantly reinforces that there's a background rate of all of these types of risk factors in the population. So, for example, loss of function variants, de novo loss of function variants, which result in literally the loss of function of one copy of a gene, are seen in about 15% of people with ASDs and about 9% of people in the population. So when one does a clinical genetic examination of someone with ASDs or neurodevelopmental disorder, it's important to recall that not every variant found is causal or damaging, because those variants can also be found in people who are disease-free. We call that a background rate. And one of the hallmarks of ASD genetic studies so far, particularly those focused on these de novo variants, is that people who have ASDs and have low IQ have more de novo variants that create risk than people who have ASDs who have high IQ. So this blue line here is the association between IQ and autism and the rate of these de novo variants from risk contributing categories, where this is something like 25% of cases have them, and this is 5%. So this is the average rate over about. And then you see up here, people with ASDs and intellectual disability have de novo mutation rates that are quite consistent with intellectual disability itself. And people over here who have ASDs and have high IQ have de novo rates that are much more consistent with the general population. And this is one of the first strong indicators from genetic studies of heterogeneity within a behavioral condition. Now, if we think about that in terms of other epidemiologic patterns and the increase in autism, what does that mean? So a lot of you also might have heard of the relationship or an observed relationship between the age at which people have children and that child's risk for autism. There's particularly been a lot of talk about the relationship between how old dad is and the probability of a child developing autism. And this is just a meta-analysis of a bunch of different studies. And what they tend to find is that if you compare dads who have a child at 50 versus a child at, let's say, 25, the increase in that child's risk for autism is somewhere in the 2 to 3 range on average across studies. So, having observed this, a lot of people started putting these pieces together. So there's some de novo variant risk for autism. Dads, as they get older, have more de novo variants. And then maybe there's more autism because dads are getting older. As there is a slight increase in average age of childbearing in the United States over the last 10 years. Nothing big, but slight. So maybe dads are getting older, they have more de novo mutations. De novo mutations cause autism, so there's more autism because dads are getting older. That was the hypothesis. So we now have enough data that we can actually check this out. And the answer is that's simply not what's going on at all. So the hypothesis largely became public because of this quote from a very widely read New York Times article. And so we did a little math on it. And here's the conservatively estimated 20-fold increase in autism prevalence in the US. And this bar, which is very, very, very small, is how much of an increase you would expect, even if dads had gotten 10 years older in the last 5 years, which simply has not happened. And in fact, in order to cover the entirety of this space, in order for the increased diagnostic rate, or increased diagnosis rate, excuse me, of autism in the United States to be explained by dads getting older, dads would currently, on average, have to be having their children at about 135 years. Which sounds off. So we're now able to actually begin linking the data, linking the genetic data, back to these observed patterns in the population and start unraveling what could or could not be going on. So another intriguing IQ-related observation in ASD research has been that people with ASDs, because of this intellectual disability comorbidity, a lot of people with ASDs also have intellectual disability, this results in a shift, downward shift in average IQ, as you'd expect. So IQ in the population has a mean of 100 and a standard deviation of about 15. So in classical genetic theory, we hypothesize that if there's a consistent set of influences on both autism and IQ that drives this association, that mean would be shifted down, which you do see. It's probably somewhere in the mid to low 80s instead of 100. But the standard deviation would be maintained. It'd still be about 15. You don't see that at all. You see somewhere between a doubling and a tripling of the variance of that distribution, the spread of the IQ distribution against the population. So if you just think of a normal distribution, here would be the population. And then if you imagine that I'm making it 2 to 3 times as big, looks like that. So that suggests that there are some genetic or some influences, they could be genetic or environmental, on autism risk that lower IQ and some that don't, or some that even increase IQ. And intriguingly, the latter, lat est, has turned out to be the case. So these common variants I was referring to, the polygenic influences on autism risk, those that exist in some degree in all of us, are actually positively associated with IQ and educational attainment in the population. So if you were to generate what's called a polygenic risk score, where you can put a number on everyone here, you could put a number on their relative genome-wide common variant risk for autism. If you had enough people, you'd see a significant correlation between how high that polygenic risk score is and IQ. So we actually see evidence that the de novo and common variant influences on autism risk, but also a lot of other phenotypes might be acting in different ways, which is important and very interesting. So while we see this very strong association between the same de novo variants that are associated with autism risk and things like intellectual disability and epilepsy, we don't see that at all with the common variants acting collaboratively, where you see the opposite association with IQ, and you don't see any association with epilepsy or seizures. So moving that idea forward, a lot of the research we've done over the last several years has been trying to understand how genetic variables create risk for autism, schizophrenia, and other behavioral conditions by trying to better understand the impact they have in everyone, not just people who are diagnosed with a psychiatric disorder. So what is the relationship between the genetic risk for ASDs that we all carry in some degree and our behavior and our thinking? Because we think those types of questions could help us greatly improve our understanding of the model through which genetic variables create risk. So going back to this, we have two types of things we're looking at again. We have the common variants working together, continuously distributed in everybody, and the de novo variants, which we also all have usually in some degree, but are less common in terms of carrying one that creates a strong degree of risk for a neurodevelopmental disorder. And we're interested in this idea. When we think about psychiatric diseases, we typically just think cases and controls, where you have a bunch of people we call cases, and we treat them like a singular mass, and then a bunch of people we call controls, and we treat them as a bigger but also singular mass. When, in reality, there could be a lot of things like this going on, where there's some underlying distribution of a trait in the population, and we're just arbitrarily drawing a line somewhere on it and calling these people cases and calling these people controls, and in so doing, losing a lot of valuable information. So people have an easy way of thinking about this sort of thing for height, which this is, or IQ, which we measure quantitatively, but a harder time for yes-no diagnoses like autism. But people have in fact thought that way for a long time. It was really originally considered by Leo Kanner, who first defined what we now call autism in 1943, and he paid a lot of attention to phenotype he saw in the parents. So he noted that he observed what we now refer to as subthreshold traits of autism commonly in the parents of this initial group of children he was describing. So that observation, which he incidentally hypothesized was due to innate factors rather than anything caused by the behavior of the parents, has led to many years of research on the relationship between clinically significant behavior problems and subthreshold traits, or traits of those behavior problems that exist on some sort of continuum. In the case of autism, they're often referred to autistic trait scales. And I think the easiest way to imagine it is just to imagine a form that's filled out by a parent, and they basically comment or provide a number to their child's ability in certain areas, including social communication. So does the child make eye contact during conversations? How do they do it making friends? Can they do certain things verbally, et cetera, et cetera? And then since you put a number to many different items, instead of getting a yes, no, what you end up with is something like this, where you get a continuous distribution of impairment or ability, or you can phrase it in many different ways, but the general idea is that down here, people are having an easier time with social communication behavior than people up here. The average score of people with autism on this measure, the social communication disorders checklist, is way up at the top, as you'd expect. But the key difference between just cases and controls in this approach is that we can recognize that these people, who are almost uniformly controls, these people aren't doing as well as these people. And then even within the cases, you can see variation in ability. And not all these people are cases, but certainly a greater fraction. These people are doing worse than these people. Okay. So a study we did recently tried to explicitly link the genetic influences on autism risk, both common, polygenic, and de novo, with those on this continuum in the population. And we estimated something called a genetic correlation, which is an estimate of the degree to which the genetic influences on two traits are shared. So a genetic correlation of 1 suggests that two traits share all of their influences. A genetic correlation of 0 suggests that two traits share none of their genetic influences. And what we found is that these common polygenic influences on autism risk share a lot of genetic, ASDs share a lot of their common genetic influences with typical variation in social communication ability in the population. So we used one of these scales, one of these trait scales, in a population from England, 8-year-olds, and we linked the genetic influences on social communication differences in kids in the general population to the genetic influences on yes, no, do you have autism? And you can see, or we did see, these dots just show the strength of the correlation between some things. These dots show that correlation between autism and typical variation in social communication ability in everybody. And these dots show the relationship between autism and schizophrenia, depression, and bipolar disorder. So what we found is that there's at least as strong or stronger relationship between autism and the way everyone varies in their social behavior, and autism and several other diagnosed neuropsychiatric disorders. There's just strong evidence for a relationship between what we consider to be typical variation and being a case. So we did the same thing with the de novo variants. We were interested in whether there was some sort of clear cut point between being affected or unaffected if you carried a de novo variant. Specifically, the question of whether if you end up with a de novo variant that creates risk for an autism spectrum disorder, does it either give you ASD or leave you alone, or does it shift you down in terms of a bunch of different traits that could be relevant to getting a diagnosis? And one way to do this is to look at people who have and have not been diagnosed with ASDs and who have de novo variant information on them. And there's one such study called the Simon Simplex Collection, where families were recruited because they have one child that's affected with autism, only one. That's why it's called a simplex collection. It means there's one case in a family. And they also studied a sibling who was unaffected. And they were measured on lots and lots of different things. One of them was a scale called the Vineland, which is a measure of something called adaptive functioning. And that includes social ability, communication ability, and things called daily living skills like can you tie your shoes, can you brush your teeth in an appropriate age. And because there's this social communication component, kids diagnosed with ASD have a downshifted Vineland distribution. So on average, they're doing worse in terms of functional behavior than their siblings who do not have autism. That's not unexpected. And then here's the siblings in blue. And they sit right at where one would expect the population level to be in terms of their distribution. This purple area captures overlap between those two distributions. So the people in this purple area are the cases with the highest functional ability and the controls with the lowest. And it's important to note that they overlap. And so we looked at that overlap in terms of people's rate of de novo variations that contributes to risk for autism. And what you can see is that when you look at that space where both cases and controls occupy, that is occupied by both cases and controls, they don't have distinguishable de novo rates. So there isn't a line phenotypically, there isn't a line in terms of behavior where you can clearly say, oh, you're a case or you're a control. There also isn't a line in terms of genetics where you can clearly say you're a case or you're a control. So mapping that back to the diagnostic expansion and change in practices and prevalence, as we diagnose more people, we're likely moving this average back. So we're moving back into the population part of the continuum as the prevalence of ASD goes up. And that's going to introduce some changes and allow some things to remain the same. As we move back here into the higher functioning range, as is often referred to, there will be less intellectual disability. There already is. And from that, we know that on average, de novo mutations will have a lower average contribution to autism. We also know that there's a link between lots of different genetic things that create risk for ASD and population variation of the traits. So those things will still matter if the ASD diagnosis continues to get more common. But these changes have to be taken into consideration as we plan and interpret our genetic studies and hopefully move soon into better understandings of heterogeneity within ASDs. So with that, I'll be happy to take questions, in particular thanks to Mark Daly and then Emily and Dan in my lab.
If people want to come down to either microphone with questions, that would be great. I'd love to start out, Elise, with a question, just juggling the microphone, that we got through Twitter. And the question is heritability the same in higher and lower functioning populations?
To the extent we can look at it now, yes, but we might not have enough people to tell if there is a difference. The number of cases that are needed to look at that question is very large. And so for the moment, there isn't a strong or obvious difference in terms of heritability as we can estimate it. But as our samples get bigger and bigger, we'll have better, what we call statistical power to look at the question.
Thanks for a lovely talk. I know nothing about autism, so forgive me for a moment. I was struck by your chart that looked at the de novo, or in a sense exogenous mutations in relation to IQ. And I guess my question is two-part. One is, forgive me for a moment, are we calling autism a disease or an illness or a condition? Because aren't there advantages to the other side of that line, the lower side, and maybe selection advantages for society in relation to those who don't have a lot of de novo changes, but are actually very bright and very focused in whatever way? I mean, is there an advantage, a selective advantage to having that group of people as part of the population? In other words, it's not seen necessarily as a negative condition, but potentially a positive condition.
Yeah, it's a complicated question, but one that we think about a lot. So I guess first, in terms of the population that receives a diagnosis, since impairment perceived by either that person or their family is a necessary component of the diagnosis, I think it's important to bear that in mind. So there is impairment associated with an ASD diagnosis, kind of definitionally. The flip side to the genetic correlation analyses, for example, the positive genetic association between common variant risk for ASDs and IQ, and what will likely be several other cognitive variables, does suggest that there are some advantages to at least relatives of those traits, genetic or biological relatives, and that we need to start thinking both in terms of the costs of having certain symptoms and kind of the potential benefits of those biological relatives when we're trying to understand genetic architecture and why certain genetic variables persist in the population.
So I have a question about, so I've read your paper on female protective effect, and I just have a question. Do you think that sex differences would have an effect on the de novo variances?
Do I think that
sex differences would have an effect on the de novo variances if you split like this data apart?
Yeah. So there's a strong sex effect on de novo variants, specifically the rate with which they're seen in cases. So I don't have slides with me on that, but across all categories of contributing de novo variation, you see an excess of those variants in female relative to male cases. Though that's actually mathematically inevitable in some senses, because I didn't talk about sex differences at all, but we can talk about it a bit now because I think we have time right now. So autism is diagnosed 4 times as commonly in boys compared to girls. And there's variation in that ratio. In terms of very low functioning ASDs, the ratio is usually 2 to 3 to 1. In terms of high IQ ASDs, it's astronomically male biased. But regardless of that, across whoever has the contributing de novo event of interest, you still see this female excess of the carriers. And that relates to the fact that there's actually a mathematical relationship between the effect size of a variant and the overabundance with which it would be seen in female cases. And something that you can model to actually help estimate the effect size of a de novo variant class is the extent to which it's seen more commonly in girls than in boys, because as the ratio of female to male carriers increases, the effect size of the variant class increases. Is that?
Hi. I was wondering if there have been any retroactive comparative studies done on the 2 different criteria for diagnosis, the original criteria and then the expanded criteria, looking at, say, longitudinally looking at individuals that weren't diagnosed the old way but have been diagnosed the new way, and then looking at the maybe two different spreads of genetic statistical data according to those 2 types of diagnosis.
Yeah, I don't think a study that effect has been done, but is becoming increasingly possible. So do you mean by the old way and the new way basically just across the range of the diagnostic expansion? Yeah. So there are now kind of data sets in which people can start to look at those questions. There are, for example, there's a wonderful resource in Denmark in which the Danish population retained neonatal blood spots from heel pricks at the birth of a baby from most babies born from the early 80s on, almost all babies born from the early 80s on. And the Broad has genotyped every case of autism born since that time in the Danish population. So from that and moving forward, you can actually start to consider these differences across birth cohort space. And I think that'll be something that a lot of people get into rather quickly now that the data are available.
Hi there. So as we have a better understanding of the polygenetic risks involved, what I'm wondering is, where do you see prescriptive recommendations? Do you see there's a role for any, or is it just that we understand the risks better and not make prescriptive recommendations on who pairs with who?
Prescriptive in terms of measuring people for their genetic risk for ASD and then making suggestions to them like who you have babies with.
Yeah.
All right. Hopefully not. So hopefully not for a lot of reasons, but at the moment there's a just functional prevention of that, and that polygenic risk scores are simply not predictive of who will or will not get autism or of most other particularly meaningful outcomes. They're associated strongly statistically with a lot of outcomes, but they're not predictive. And for example, polygenic risk scores for schizophrenia are currently the most useful in psychiatric space because the schizophrenia sample sizes are the biggest, and that yields your most useful risk score. While the schizophrenia risk score very strongly distinguishes cases and controls, it is terrible at telling you who in the general population will be a case. So for that same reason, even once we get to the type of sample sizes that are seen in schizophrenia, we will not have great utility to do that, at least for the foreseeable future. It's also important to note that there are companies that claim to have SNP tests or common variant tests for autism risk, both for the general population and for people who, for example, already have one child affected with autism. There's currently no reason to believe that those work, though they can be sold in an unregulated way directly through a physician. And the kind of regulatory environment hopefully will also soon catch up to corporate claims to the contrary.
Why the 48% similarity monozygotic twins?
In schizophrenia? Yeah, so I had a slide at the beginning that showed something like 50% concordance, we call it, when two twins are both affected. That's called concordance, between identical twins with schizophrenia and something that I think was vaguely 16-ish% for the non-identical twins. Yeah, sounds about right, yeah. So the reason that 48 or that 50 is not 100 is just because none of these disorders are entirely genetic. They have very strong, and in the case of schizophrenia and autism, predominant genetic contributions to risk, meaning genetic factors create the majority of the risk. The environment, however, is very important. It's just harder to study, and as such, scientists have been slower to identify specific environmental risk factors. But the reason the twins aren't always concordant is because they share all of their genetic material, but then they end up not sharing a lot of other things once they are born.
One of your earlier slides, you were showing us the red bars with the ascending blue bars, one indicating people who suffer from ASD would have an intellectual impairment, and those who don't see that kind of diagnosis. I was curious why there was such a strong difference between a population like Utah and New Jersey. How much of that is predicted or explained by the diagnosis process versus the underlying genetics?
I don't think it is in any meaningful way the underlying genetics. Were I to take a guess, which I'm kind of hesitant to do since this will end up on YouTube, but we can do it anyway. There are a lot of social and political factors that relate to estimates of any disease prevalence in any population. A couple of them that are likely, but we cannot say for sure, relevant to this, is the Utah and New Jersey thing is curious in part because Utah and New Jersey hosted the first large prevalence studies of autism in the United States. And there's likely a relationship between the success with which people in Utah and New Jersey can find autism cases and the surveillance systems that were set up for those studies. Then there's factors that build on it. For example, states that have very good resources for children with developmental disabilities and their families often have families with affected children move to them. So you end up with this building effect over time. But the short answer would be I do not think major genetic differences between people in Utah and people in Arkansas are relevant here.
So you've mentioned bipolar and schizophrenia being factors. Could you also comment just with your data sets, perhaps what you found about ADHD? Because diagnoses, those seem to also be on the increase as well.
Yeah. So at present, the relationship between ADHD and autism is a tad curious in genetic space because, as you mentioned, there's a very strong comorbidity. It's often many people with ASDs also meet criteria for ADHD. And in twin studies, it's been repeatedly shown that there is genetic overlap between risk for ASDs and risk for ADHD. Current studies of genetic overlap that use measured common variant data haven't found that for reasons that are unclear. But those studies have also had reasonably small samples of people with autism and ADHD each. And in this case, at this point in time, we refer to a small genetic study of that sort as being less than 5,000 people. And that might change in the future. So a nice thing to note about ADHD is that trait is actually also very strongly hypothesized to exist on a continuum, and perhaps even a more evident continuum than autism. There's also genetic data to that effect emerging as well.
More questions for Elise?
I think there are also cases where somatic mutation during early development drives the autism. But I think there's no really tool for screening out somatic mutations, which would be only possible if we can gain great tissue samples. So do you think there would be a high proportion of somatic mutations driving autism in the full population?
Yeah, he asked whether somatic mutations have strong relevance to autism. Somatic mutations are mutations in genetic material that occur after the formation of a germ cell, basically. So people vary in their opinions of this. My guess is not that much, but those data are not widely available. So I can speak with you afterwards. There are a bunch of people whose work you can look at who have started to think about this. Yeah.
I was wondering, you had on one of your slides, you showed that there was a large difference in autism that was diagnosed in the 1990s compared to present. And I was wondering if in part that might have been caused because there might have been less ways to diagnose autism.
Sure. He asked if whether the fact that more autism is being diagnosed now than in the 1990s might reflect fewer ways to diagnose it. Do you mean kind of access to clinicians or services or something like that?
Yeah, and also perhaps less methods to diagnose.
The methods are actually pretty consistent. A diagnosis of autism still requires a structured interview from a qualified clinician, and it actually hasn't changed that much in quite a long time. So in terms of more access to people who can make a diagnosis, I think it's a bit complicated. So on the one hand, there might be more access to services in certain places, certainly. On the other hand, a lot of the diagnosis awareness issue that relates to prevalence spreads far beyond people who are technically qualified to make an autism diagnosis. So you'll have, for example, every pediatrician being more aware of what autism and autism symptomology is now than on average they would be 30 years ago. And that spreads out to people like teachers, it spreads out to occupational physical therapists, where there's generally greater awareness and greater discussion. So when people see symptoms in a child that could be consistent with a social communication deficit, there's just a markedly increased probability that that child will end up being clinically evaluated for ASD.
Hello. I'd like to know if you happen to know if there's any research in this area internationally, or is autism spectrum disorders found in other countries and other cultures?
Yeah, it's found all over the place.
Yeah.
It's most commonly studied in terms of genetic research communities in the US and Western Europe, but all genetic research is most commonly conducted in the US and Western Europe at present, but that's rapidly changing. The Stanley Center at the Broad has a lot of large new collaborations with international partners designed to study autism and other major neuropsychiatric illnesses like schizophrenia and bipolar disorder. We've actually specifically begun a collaboration designed to study autism in sub-Saharan Africa, and we have fantastic partners with which to do this. And one of the immediate, kind of an obvious influences on what we expect to see in terms of genetic data is that autism as it's currently diagnosed in sub-Saharan Africa looks like, a lot more like, autism as it was diagnosed in the US 30 years ago, because those locations have not experienced this diagnostic expansion, nor do they typically, not typically, but nor do many of them have the resources necessary to see all but very severely impaired children. So in studying autism there and intellectual disability there, we'll be doing both, you expect to see genetic profiles and phenotypic profiles that will look a lot more like the more severe forms of autism that were commonly diagnosed in the US and Western Europe decades ago.
Okay, we have another question from Twitter. Can you speak to any link between autism genetics and microbiome?
Other people can, but I can't. It's not my thing. People often guess that if you study one kind of ohm, you kind of know about the other ones, but they don't actually overlap that much. But whoever tweeted that can, I don't know, retweet you and I can give them some names.
Are there more questions? Okay. All right, well, thank you, Elise. We really appreciate it.